Welcome everyone. This video is meant to be a brief introduction to notch signaling. And as you think about notch signal transduction, I want you to associate notch with cell proliferation, cell survival, and de-differentiation. In other words, maintaining a more plastic phenotype. And as we explore this pathway, there's a couple of concepts that I'd like you to consider regarding notch signaling. The first one is sequestration of the active notch molecule. Of course, to sequester simply means to isolate or to hide away. The second is that notch is involved in direct cell-to-cell signaling. In other words, typically for this pathway to become activated, two cells have to physically touch each other. And then, of course, the third point is the end game. What are we after? Well, for notch signaling, as for a lot of the signal transduction pathways that we'll look at, the end game is gene transcription, which of course ultimately leads to protein expression. And in the case of notch signaling, these proteins will promote cell proliferation, cell survival, and de-differentiation. Now, this signal transduction pathway is one of the simpler pathways that we're going to look at. Let's introduce the key players. Well, to start off, of course, we've got notch, and the ligand for notch is a ligand named delta. We'll also discuss CSL proteins, a protein called mastermind-like, which is abbreviated MAML, and a family of proteins called Hess. So now that we've got our main players, there's not many of them, let's look at this pathway. As we said, this pathway typically requires direct cell-cell interactions. So let's draw one cell. Here's the second cell. And expressed on the first cell is our ligand delta. And the interesting thing about this is that delta is membrane-bound. And delta is the ligand for notch. So as you can see, when these two cells come together, we have an interaction between delta and notch. And what this interaction does is it slightly changes the notch configuration to expose a weakness, a cleavage site, if you will, where metalloproteases can come cut and thereby liberate what we call the notch intracellular domain. So this highlights one of the points that we mentioned earlier about sequestration, right? The active part, this notch intracellular domain, is sequestered to the membrane because it's physically bound through the rest of its protein in the membrane. So the activation process is simply exposing a cleavage site where a metalloprotease can come in and cut and liberate this notch intracellular domain, which is the active portion. Now, once liberated, what does this intracellular domain do? Well, now that we're not tethered to the cell membrane anymore, we're headed to the nucleus. And as we said earlier, the end game, of course, is gene transcription. So, as the notch intracellular domain translocates to the nucleus, now we can assemble our transcription factor complex. And in the spirit of constructing a human body, let's symbolize this transcription factor with a construction machine. How about a bulldozer? So this notch intracellular domain is going to recruit CSL proteins and the mastermind-like protein. And now we have a functional ternary protein complex that's able to transcribe genes. And for this transcription factor complex, the primary genes that we're going to be transcribing are Hess family genes. Importantly, Hess proteins are typically repressors. And this describes how Hess can, for example, block differentiation to maintain a de-differentiated state. And sometimes during development, certain Hess proteins, specifically Hess1 and Hess7, actually demonstrate oscillatory expression patterns. In other words, they turn on, then they get turned off, and then they turn on, and they get turned off, and this has to do with negative feedback. I'm going to mention this in particular because of its role in somatogenesis. So now that we have the general picture of what notch signal transduction looks like, what can happen if notch signaling is perturbed? Well, one example is spondylocostal dysostosis. This can occur, for example, with mutations in notch2. And what you can see from the X-ray and the cartoon are ribs that are not formed or even missing, vertebrae that don't form properly. Sometimes you can call these butterfly vertebrae because they don't close properly. And of course, the spine itself is a direct result of somatogenesis. And we talked about the role of oscillating notch proteins in somatogenesis. And this just provides one example of the importance of proper notch signaling in human development.